Hello, everyone, and welcome to today's webinar, Demystifying Exosome Isolation, Best Practices in Centrifuge Selection Strategies. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, please visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box at the left of your screen and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. And with that, I'd like to now introduce to you our speaker for today's webinar. Marzieh Mousavi, Master of Chemical Engineering, Product Application Scientist, Thermo Fisher Scientific. For a complete biography on our speaker, please visit the Biography tab. Welcome, Marzieh. You may now begin your presentation.
Hello everyone, and welcome to our today discussion about exosome isolation techniques and their best practices. My name is Marzieh Mousavi. I'm Product Application Scientist at Thermo Fisher Scientific. So there's a significant amount of ongoing research on extracellular vesicles, specifically exosome centrifugation technique as one of the most popular separation that's recognized as a gold standard need to be fully explored and understood. Today we'll start with an introduction to exosome and the overall research workflow, including existing isolation techniques. Then, before reviewing isolation protocol using extracentrifugation, we'll learn about the centrifugation principles. And finally, we'll wrap our discussion with thermoscientific solution and that followed by question and answers. Exosome. Exosomes are small membrane-bound vesicles that's secreted by cells and are a subset of extracellular vesicles. Extracellular vesicles, or EVs, are membrane-bound lipid vesicles that's released by most eukaryotic cell type as a means of intracellular communication and cellular waste. EVs are classified into exosomes that range between 30 to 150 nanometer and are produced through the endocytic pathway. Microvesicles that range from 50 to 1000 nanometer and produced through outward plasma membrane budding. And the largest EVs are apoptotic bodies that are that range that more than 500 nanometers. So due to differences in the biogenesis pathway and originated cells, the molecular content of our EVs are different. So today we will focus only on exosomes that range between 30 to 150 nanometers. The history of exosome can be traced back to the period from 1960s to 1980s that think that exosome is a junk molecule. In 1987, R.M. Johnson was the first scientist that coined the term exosome to these vesicles. In late 90s, researchers began to recognize exosome as important carrier of molecular cargo, including protein and RNA. In 2007, Valadi et al. Demonstrated that exosome can transfer functional RNA molecules between cells. In 2013, the Nobel Prize was jointly awarded to 3 scientists whose their work significantly impacted our understanding of cellular communication. In 2018, FDA approved the first exosome-based product, ExoFlow, as a human cell and tissue product. So in 2020, during the pandemic, the first clinical trial of exosome-based COVID-19 treatment started, and today we have 328 registered exosome-based clinical trials. As you see that the number of research projects involving exosome is rapidly growing. So exosome researchers continue as a treasure hunt to new discoveries and still working on finding a standardized exosome in terms of isolation and characterization technique. Therapeutic application, exosome can serve as biomarker for diagnosing and predicting and monitoring cancer by analysis of the molecular content of isolated exosome, like proteins, nucleic acid, and lipids. Exosome also has immense promise in the field of therapeutic. For instance, in drug delivery field, exosome can serve as a drug delivery platform that minimizes side effects and can maximize treatment efficacy. In regenerative medicine, exosome has the capability of regenerative properties and can promote tissue repair and regeneration. Researchers are also actively engineering exosome to enhance immune response against infectious disease, cancers, and autoimmune diseases. Exosome also even finding application in vaccine development by stimulating immune response and potentially enhance vaccine efficacy and delivery. The key point here is that exosomes are not just microvesicles. They are potentially game changers in the world of medicine and science. The overall workflow for extracellular vesicles, including exosomes, start with sample collection and preparation as a source of isolation. Then exosomes are isolated by available technique that we'll discuss more in the next slide. Then before the application, isolated exosomes need to be qualified and analyzed by different techniques like electron microscopy, flow cytometry, vesicle blotting, and more. But there are many challenges in the entire workflow that need to be addressed. For instance, in isolation process, there are over thousands of different combinations of how people perform differential ultracentrifugation to get their EVs prep, or sample heterogeneity is a big challenge in the exosome research workflow. Depending on the source of EVs, there can be subset of EVs. It's been shown that you can isolate EVs in two different ways and they can have two different downstream effects. Another challenge is scaling without compromising the quality. That need to be addressed and definitely need some investment on that. So from a drug development perspective, the key is making sure that what you are delivering is reproducible and quantitative, so that preclinical study can rely on the process. Also in analytical step, there are tools that can be measure the entire diameter of extracellular vesicle distribution in a high throughput fashion and recognize exosome from co-precipitated microvesicles. So the key point here is we need to have a standard and reproducible isolation technique in EVs and workflow and also exosome research workflow. These 6 main isolation techniques are developed to isolate exosome. The first one is sequential ultracentrifugation that can produce high volume of exosome and it's a scalable solution, but other biomolecules can precipitate with exosome. Gradient ultracentrifugation, on the other hand, can produce high purity of exosome, but the process is long. Ultrafiltration technique is quick and easy, but the yield is low and filter membrane will clog in this process. Size exclusion chromatography also showed minimal structural damage. They are reproducible technique, but exosome that prepared by size exclusion chromatography displayed wider size distribution. The other technique that is based on the polymer precipitation, and most kit reagents developed based on that, they are easy to use, fast, and we have high yield in this technique. But the problem is water-soluble material like nucleic acid, lipoproteins, or even virus reported that can precipitate with exosomes. Another technique, which is based on the immunoaffinity, this technique is single-step, easy, but the problem is it only can isolate a specific population of exosome that has that specific antibody. And also the ocean buffer affect the biological function of the exosome with this technique. So you see that many various techniques are developed to isolate the exosome, and each technique has some strengths and weakness. Among all developed technique in exosome isolation, according to the publication and Exocarda database, 72% of publication use ultracentrifugation technique, and 6% use ultracentrifugation plus density gradient technique. So ultracentrifugation can be adapted for various sample types and volume. It's scalable. Isolation or protocol is established since 1987, and by combination with density gradient technique, high quality of exosome can be isolated, and most research lab has access to ultracentrifuge instruments. So that's why ultracentrifugation recognized as a gold standard technique for exosome isolation. For those who are not familiar with centrifugation technology, in theory, particles sedimentation can be achieved with the help of gravity, but that would take very long time depending on the mixture. So the centrifuge is a device that accelerates gravity and separates particles from a solution through the use of a rotor. So in biology, the particles are usually cells, subcellular organelles, large molecules, genetic material, or viruses. So in centrifuge, we usually state the speed based on the RPM, round per minute, but relative centrifugal force, RCF, is a universal currency of centrifugation, of centrifuge application that must be used for particle transfer. So RPM and RCF can be converted to each other by knowing their radius, the rotor radius. Three different techniques have been developed in centrifugation. The first one is differential centrifugation. This technique separates particles based on their size and density differences. Heavier particles sediment first. The second technique is isopycnic or equilibrium centrifugation, and that separates particles based on the density in a density gradient medium. So the sample is loaded onto the top of the gradient, and during the centrifugation, particles move within the gradient until they reach the region which their density match of the surrounding medium. The last technique, or the third technique, is the rate zonal centrifugation. In rate zonal, particles separate based on the sedimentation rate. So the sample is layered on top of the density gradient medium, that the density is lower than the particles. And as the centrifuge operates at a constant speed, particles of different size or mass sediment at different rates and form distinct band within the gradient. So unlike isopycnic ultracentrifugation, because of the concentration of medium in this type of ultracentrifugation is lower than all of the sample component, all the insoluble particles can be pelleted at the bottom of the tube after prolonged centrifugation. So this technique is very time sensitive, and the centrifugation time need to be carefully selected. So most exosome isolation technique developed based on the differential centrifugation and isopycnic centrifugation. We have a few publications that use rate zonal centrifugation, but as it's very time conservative, few papers or few research use this technique. To understanding what G-force can isolate exosome vesicles, we can compare it in a chart of G-force for different biomolecules according to the size and density of different types of EVs, apoptotic bodies that are large molecule sediment at low G-force, like around 2000 G. Large microvesicle sediment at about 10,000 G-force, and exosome vesicles precipitate at 100,000 G to 150,000 G-force, which is in a range of ultra-centrifugation speed. But as you can see in the graph, if there are other components like LDL, HDL, and even VLDL can co-precipitate with exosome. So with that, exosome isolation by ultracentrifugation technique, first established by serial centrifugation steps. Three main workflows are developed with ultracentrifugation techniques in most literatures and research publications. So in all of them, 3 steps of low G-force centrifugation are applied to remove cell debris and apoptotic bodies and large microvesicles. Then supernatant goes to workflow 1. That supernatant is centrifuge at 100,000 G for 90 minutes, exosome pellets collected and resuspended in PBS, and centrifuge one more time at 100,000 G for 90 minutes to be concentrated. So this protocol is simple and fast and scalable, so we can produce large volume exosomes, but the other biomolecules, if presented in the source, that has same size of exosome, 30 to 150 nanometers, can be co-precipitated with this protocol. So this is ideal technique for less complex like cell culture media and when we need large volume of exosome for our studies. So the workflow 2 that leverage differences in density and added 1 step sucrose gradient to remove impurities. So this workflow is pretty like the first workflow, but the only difference is after 2 steps of low G-force centrifugation, supernatant is loaded on top of the 30% sucrose gradient that has same density of exosome vesicles. So after 90 minutes centrifugation at 100,000 G-force, only exosome will be pelleted into the sucrose gradient. Then PBS will be added to the sucrose gradient, and we can repeat the centrifugation to concentrate the exosome. So this technique is takes the same amount of time as the workflow 1, and it has high recovery rate, and exosome vesicles reported has the high purity, and the morphology of exosomes are intact. So the last protocol that we'll see in many literature actually is the first protocol with extra step of purification with density gradient to remove protein contaminants. So layer of biocompatible medium with varying density like iodaxinol or sucrose that covers the range of particle density in the sample is placed in a tube, and that gradient needs to be gradually decreasing density from bottom to the top. Then isolated exosome from protocol 1 loaded onto the top of the layer and followed by a prolonged period like 16 hours. So protein aggregate concentrated at the bottom of the centrifugation tube, and exosome remain in the layer of probably medium between 110 to 118 gram per ml. So then eventually the layers contain exosome centrifuge one more time at 100,000 G-force to be concentrated. So this technique provides the purest vesicles among all those techniques not centrifugation, all of the available technique in the market, and it's ideal for complex sample like blood, plasma, and for the functional study of exosome. But the drawback is process takes a long time in this technique. Although all these protocols are established, but we need to consider several important factors once we're designing on experiments. For instance, sample collection and processing is very important that impact the exosome contents and biological functions. Always choose the recommended preparation techniques in literature. For instance, once we want to isolate from whole blood, blood plasma, EDTA treated need to be considered. Or in cell culture, we need to use serum-free medium. And always record all the details about the sample collections like age, time, gender, place, and how it treated. Another parameter is always choose the right platform based on the sample complexity. Complex fluids such as blood plasma need purification steps on top of the isolation step, like for workflow 3 that we learned about that. And then select the centrifuge and rotor based on the sample volume, and always choose the right rotor can optimize our protocol. For instance, if we want to pellet the exosome, fixed single rotors can provide us better results, and swinging bucket has higher resolution for density gradient technique. And reproducibility is the key of success in this workflow. So to do that, we need to record all the details like centrifugation parameters, G-force, rotor rag factor, K-factor, temperature, acceleration, deceleration. And also we need to record all the sample collection or process we did on that, and characterization and quantification details, and the normalization technique that we used. Thermoscientific has broad range of centrifuge available in exosome isolation technique in both centrifugation that need low G-force and isolation and purification that need ultra G-force. We also have versatile range of rotors and bottles to accommodate small volume to large volume of samples. For more information, please scan this QR code that take you directly to all the available solution in Thermoscientific. And if you are interested, you can contact our representative for more information and detail. In conclusion, extracellular vesicles, including exosomes, application growing rapidly. Regardless of the application, a standard and reliable isolation technique that can reproduce high quality of exosome is essential in this field. Ultracentrifugation, that's recognized as a gold standard of exosome isolation, is well documented, and it provide a scalable solution to the market that can produce high quality exosome for all application. If you are interested, you can scan this QR code, and it gives you access to our 2 application notes that we published. Thank you for your attention. We're now ready to address any question you may have.
All right. Great. Well, thank you so much, Marzieh. All right, we're now going to start the live Q&A portion of the webinar. So to our audience, if you have any questions that you would like to ask, please do so now. Just type in your question to that Ask a Question box and click Submit. We'll answer as many of your questions as we have time for. All right, so we've already got a few here being submitted into the audience or into our side, which is great. So I'll go ahead and start with this one. This first question asks, after isolating exosomes, how can I wash them?
So the best way to wash the exosome is adding PBS, which is a salty solution, and it doesn't affect the exosome function. So add that, and then you can centrifuge it during the concentration. Once you add it, you centrifuge it another time at 100,000, and it's basically washed your exosome vesicles.
Wonderful, thank you. All right, another question here. How can I concentrate exosome pellets?
So if you look at all those protocol, the last step is adding PBS, and we need to centrifuge it 100,000. So then we retrieve all the supernatant and let about 1 ml or 2 ml of PBS with the floated into that. So that actually provides you the concentrated vesicles.
Great, thank you. Next question. I work with a biosafety level 3 pathogen. At which step of differential centrifugation is suggested to filter the supernatant through 0.22 filter?
Yeah, that's a great question. So there's a lot of protocol that recommended using 0.22 microfluidic filter, and it's right after 3 steps of low centrifugation. We can use filter before starting our ultracentrifugation process.
Great, thank you. Another question I have for you. Even after following all these kinds of centrifugation techniques, there still remains some sort of protein contamination. Is there any extra purification methods or steps to purify the isolated exosomes?
So in our density gradient platform, which is workflow 3, that if you needed high purity of exosome and get rid of all the lipoproteins or proteins, so you can load it over the density gradients medium, and then that provide you the best quality based on the literature, best quality of purified exosome. So my answer will be using density gradient technique.
Great. Next question, what is the best way to store my exosomes?
So if you want to store for a short time, like 1 week, you can store it at 4 degrees C. But if you plan to store it for a longer time, like a month, minus 20 degrees C and minus 80, I would say, is the temperature that recommended to store exosome. But if you want to use it in different time or you have a high concentration, the best practice is aliquot your exosome product, and then to avoid any thaw and cycle times, multiple thaw and cycles. And because multiple thaw and cycles would affect your exosome function.
Great. All right, well, we do have time for a few more questions here, so we'll continue. This next question asks, after isolating exosomes, what are the main QCs to quantify or qualify exosomes?
So for quantify exosome, the best technique is nanoparticle track analysis that give you the distribution of size. Western blotting is another technique, and flow cytometry, I would say, is one of the technique that used. And more for TEM that gives you full image of your exosome vesicles.
Great. Another question I have for you here. What rotor for WX Plus do you suggest to maximize output? Like 1,000 ml per run of fluid containing exosomes?
So 1000 ml. So I would say our carbon fiber rotor. So I don't have it here in the slide, but I definitely can provide more information on that. So our carbon fiber has ability to spin about 400 ml with WX, but you need to at least 2 or 3 run to give you that 1000 ml.
Great. Next question, can isolated RNA from EVs be tested for integrity, as in RANs? Is there a recommended method of integrity testing for exosomal RNA?
Okay, so can you please repeat that question?
Yes, no problem. Can isolated RNA from EVs be tested for integrity, as in RANs? Is there a recommended method of integrity testing for exosomal RNA? Excuse me.
So I don't have answer to that, so I can give you back with the answer. Yeah.
No problem. Perfect. That sounds great. All right. Next question here. What is the best technique to isolate RNA from exosomes from plasma?
So for the RNA, so after you loaded your isolated your exosome, you can isolate it or RNA based on the routine technique in the market. So, but you need to first isolate your exosome vesicles.
Great, thank you. All right, we have time for just a few more questions here, but we will continue. We're getting some great questions in from our audience. Next question, can exosomes be detected with LRS for TESA?
Sorry, I can give you the answer with this question. I'm not expert on that techniques.
No problem, that is okay. All right. Would exosomes be frozen in PBS, or is there another diluent that should be used?
No, PBS is one of the most common solution that recommended to use for storing exosome at -80.
Great, thank you. All right. Another question, how can I dry the isolated exosomes, and what could be the maximum temperature that exosomes can withstand?
So exosome need to be isolated in all the process. It recommended 4 degrees C. And because the high temperature will affect the exosome integrity and exosome bifunction, so we recommended set the temperature at your centrifuge and instrument at 4 degrees C, and before loading of your sample, let the centrifuge and rotor be stabilized at 4 degrees C to avoid any high temperature during the centrifugation process.
Wonderful, thank you. All right, looks like we have time for one more question here, so we'll wrap up after this one. Speaking of angle rotor, will angle rotor be actually suitable for the task because of, well, the fixed angle?
Yeah, so fixed angle or so for the pelleting in the workflow 1 that we use the differential centrifugation, fixed angle is one of the type of rotor that we recommended, but in the density gradient technique, we'll recommend that using our swinging bucket that will give you the higher resolution.
Wonderful. Okay, great. All right. Well, this will conclude our session for today. So before we wrap up here, I want to thank you, Marzieh, for your time and your important research. But before we go, do you have any final comments for our audience?
No, thank you. I would like to thank you for attention. So if I wasn't able to address 2 questions here because I'm not expert in the analytical, so I'm expert in the centrifugation. So I will have your information. I'll come back with you with the answer. Yeah.
Wonderful. All right. Well, thank you again, like I said, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Now, before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.
Thank you. Bye.